Hello and welcome. We're going to wait a minute or two for the attendees to join online. Okay, let's go ahead and get started. Welcome to the Psychiatric Genomics Consortium Worldwide Lab Meeting on September 27th, 2019. Thank you for joining ahead of the conferences and during grant season. I know that at least 2 presenters are in total crunch time for the October 5th deadline for NIH, so I appreciate making time for this, for you guys presenting and for all of the attendees. Today we have insights into the genetics of suicide from a group of collaborators within the PGC, the International Suicide Genetics Consortium, and the PsychEmerge Consortium working with EHR data. We have Professor Hilary Coon from University of Utah, Assistant Professor Anna Docherty from Utah and the Virginia Commonwealth University Institute for Psychiatric and Behavioral Genetics, VIPBG. Assistant Professor Doug Ruderfer from Vanderbilt University Medical Center, and postdoctoral fellow Niamh Mullins from Mount Sinai School of Medicine. Their talk titles are shown on the second slide. Dr. Docherty, could you advance the slide, please?
Excuse me, it's not advancing.
Oh, you had to hit escape yesterday and then it started working.
Apologies.
No problem.
Okay. There we go. There we go. Sorry about that.
And you need to share screen again, I think. Okay, the talk titles are shown on this slide. We're planning 4 short talks, 10 to 15 minutes, and we hope that there's time for a short question and answer period at the end of each talk. And then, according to plan, we'll have 5 to 10 minutes at the end for questions or any broader discussion. My plan is to look on the attendees list for people who raise their hand. If you raise your hand to ask a question, then I can unmute you and you can ask your question directly with the panelists here. So we'll see how this works. Typically then also, you can send questions, you can type your questions into either the Q&A button after pushing the Q&A button at the bottom or the chat button, and we'll read your questions aloud and then answer them. September is Suicide Prevention Month. Is that so, you ask? Well, every month should be Suicide Prevention Month. And so we focus on suicide biology and genetics. And this is Hilary Coon.
Thanks very much. It's a pleasure to be able to present to you, and we're absolutely delighted that suicide is now sort of moving into the mainstream of psychiatric genetics. And this, of course, it's been sort of on the fringe, but this hasn't been because it isn't important. We never have any trouble with our impact statements in our grants. So suicide is certainly a huge public health crisis. 10th leading cause of death in the US. In 2017, over 47,000 people died by suicide, and this number is over 800,000 people worldwide. And attempts are much more common than suicide deaths. So for every suicide, there's 25 to 30 attempts. So a big worldwide problem, a growing problem. I'm going to give you a little bit of overview of suicidal behaviors and also then suicide death. And our talks are sort of spanning the spectrum of those two outcomes. So certainly the problem is that attempts are increasing, and our prediction of attempts is pretty poor at this stage, which means prevention is really challenging. And a possible solution to this is really lying in genetics. So the heritability of suicidal behavior from twin and family studies is up to 55%, which suggests some promise for using this type of data to really help us in prediction and prevention. We have an opportunity though, because most of the prior genome-wide studies have been within specific psychiatric diagnoses, and actually most of them really have not been large enough to identify any replicated genome-wide significant loci. So the time is really right for the studies, using really large population samples and also the very large psychiatric samples within the Psychiatric Genomics Consortium, and you'll hear about both of those efforts from our colleagues, Drs. Ruderfer and Mullins. At Utah, we actually are focused more on suicide death, and this is partly because of some resources that I'll tell you about in a few minutes. And I just wanted to highlight some of the differences between suicide death and suicide attempt, which we're really focused on trying to figure out some of the potential differences in genetic etiology between these two outcomes. So just on a very basic level, suicide death is way more rare than attempts. I mentioned this in the previous slide. And then if you look at another very sort of basic etiological variable, which is sex, suicide death is very male. So in the US, this male-female ratio is almost 4-to-1. For attempts, it's more difficult to count these accurately, but they're far more common in females. And this is especially in youth, where the females are about twice as likely to make an attempt. And it is also worth noting that I showed you a slide about the incidence of attempt increasing. Well, the incidence of suicide death is also increasing dramatically. It's increased about 33% since 1999. So one issue that we think about a lot is, to what extent is suicide really sort of some dramatic etiological similarity with mental illness? So many of the individuals who die by suicide struggle with mental illness, and this has often been a chronic situation. But we also know that most individuals who have a mental illness don't die by suicide. So there's some indication that suicide has some other etiology over and above just psychiatric vulnerability. So the slide I have here is actually not a Utah big extended family. This is actually from Janice Eglin's seminal work in the Old Order Amish. This is a suicide family that is one of many families that she studied for major depression and bipolar disorder. And what she found, she had records for suicide that spanned 100 years in this cohort. And of those suicides, all of them fell into just 4 of the families that she was studying. And she made a conclusion, this is in the 1980s, that suicide was significantly familial, and that it looked like there were some risks that were independent of psychopathology, or at least that spanned multiple psychiatric disorders. There's some other seminal work that's been done by Brent and Mann, published in 2005, that also suggests that there are potentially risk factors unique to suicide. I mentioned that suicide attempt is significantly genetic. These are some data from aggregated studies, twin and adoption studies of suicide death. So this outcome also looks like it's significantly genetic. You can see the identical twins much more concordant than fraternal twins. And also in the adoption studies, the biological relatives much more at risk than the adopting relatives. And from some meta-analyses of this work, the evidence for suicide death is actually similar to the heritability for attempt, is about 50%. Okay, so why Utah? Well, in Utah, we have this sort of dubious distinction of being in the top 5th or 6th for suicide rate. We dipped down to 6th recently, but still we're in what's called the suicide belt of the US. And in Utah, suicide is actually the leading cause of death for people under 25. We also have some just really great resources that make the logistics of doing this fantastic. So we have a centralized medical examiner's office. We have a medical examiner who's really interested in collaborating with us, and we've been collaborating for about 20 years. We now have over 6,200 individuals who died by suicide with DNA, and this grows at the rate of about 700 a year. So within about the next 5 years, we'll have about 10,000 individuals who died by suicide with DNA. We also can link these cases to a fantastic population resource called the Utah Population Database. This is a statewide database that includes access to extensive medical records data, demographics, genealogical records, geographical data, which allows us to look at exposures, and it's a terrific resource. Now, one of the potentially most unique aspects of this resource is actually the extended families. We have very large high-risk families in the Utah Population Database through a gift of the genealogical data from the LDS Church. And we have identified 43 very high-risk families where we've then looked for familial sharing, genomic sharing in people who are then related in these distant way. We've actually published this in Molecular Psychiatry, and we have 30 genome-wide significant regions in 10 of these families. We're following this up with some sequence data. And I listed a couple of these families below that gave us these significant regions. The people that actually have genotype data are circled in red. So we found that the genomic regions that are familial, these are challenging to follow up, because what's driving this significance is likely regulatory. It's not necessarily really easy to see, single variant, very high penetrance. So what we're doing in the meantime is actually using the familial cases in a couple of different ways to address some other hypotheses. So these people in these families actually represent cases that enhance genetic risk. They often, once we get up a couple of generations, are tied to multiple common founders. So the genetic risk is actually not just they're part of one single family. We affectionately call this spaghettigrees in the Utah Population Database, and this happens with other conditions as well. So they really represent familial enrichment. So this is also genetic enrichment, right? Because these cases share very little common environment. They're very distantly related. So what we thought we'd do is look at these cases and see if they look like they have significantly elevated risk of psychopathology by using polygenic risk scoring. And you'll hear more about the polygenic risk scores from my colleague, Dr. Docherty. But what we did was we looked at these genetically enriched cases, the European subset of them, calculated 60 polygenic risk scores for a variety of traits. And then we've just looked at the difference between these genetically enriched cases and our other suicide deaths, then again adjusting for residual ancestry effects. And we also included the polygenic risk of suicide death actually derived from our own cohort. And we use a tenfold cross-validation to do this since it's derived in our own cases. So here's what we find. The high-risk familial suicides are actually significantly elevated just for risk of suicide death. So we did not find any enrichment for any other psychopathology in these cases that are sort of selected, right, for genetic risk. They just look like they're enriched for suicide death. And we found this to be very interesting. We were kind of expecting them to be enriched for some other genetic risk of psychopathology, didn't find this. So we're also looking at these cases and beginning to see some association at gene pathway level between them, looking at rare structural variants and single nucleotide variants of high impact. These look like they're they're starting to cross-match with findings from our GWAS, which is very interesting. If you want to talk to me about that, you'll have to find me at World Congress. I don't have time to tell you about that now, but it's exciting work. And we really believe that studying suicide death is important. Of course, studying attempt is very important as well. But by studying suicide death, we can really try to get at genetic discovery for the very highest of the high-risk cases. And we can really start to look at the extent to which etiology differs between suicide attempt and death. And I just want to acknowledge a ton of people who are working on this project. It's a really big resource, fantastic resource, lots of folks at University of Utah, and then across different departments within our university, then many external collaborators, and this is growing. We're really excited to be parts of the international consortium work. And lots of folks who have supported this work.
Thank you, Hilary. I think that it's worth it to take a brief time for questions. If anyone has a question, please raise your hand, or again, type your question into the Q&A box or the chat box quickly, please, before we move on to the next presentation. Five, 4, 3, 2, 1. Okay, we'll move on to the next presentation, which is Anna Docherty.
Okay, thank you, all of you attending today, and I am going to briefly discuss genome-wide association analysis of the suicide death samples here in Utah from the incredible resources that Hilary Coon has obtained here. And this is going to be a brief talk, so you know, I'm obviously happy to provide details to interested people offline as well. So our genome-wide association study of the first genotyped suicide deaths was a case control design with 3,413 suicide deaths from Utah. These are non-Finnish Northern European suicide deaths, and 14,810 controls. And we had actually some significant challenges to the genome-wide association analysis using Utah suicide data, because we don't have controls in Utah. And so we had to take multiple steps to filter and minimize false positives in our analyses. Another challenge is that we have only about 20% of our sample has significant admixture. And so with this preliminary data set, we didn't have a sufficient sample to complete a trans ancestry GWAS, although we do expect in the next 3 years to have the numbers to do that. So I'm going to talk about the steps to minimize false positives in a moment. These are the PCA plots of the suicide deaths, the controls from Generation Scotland and UK 10K that we used, as well as 1,000 genomes superpopulations. On the left here, you'll see plots of the entire sample, and then on the right, you'll see this is zoomed in on the hinge of that entire sample there. On the top, you'll see the suicides and controls mapped to 1,000 genomes. On the bottom, you'll see just the suicide deaths and the control samples, along with those that we excluded for this analysis. And then, like I said, we did take multiple steps to filter out potential false positives. One, we jointly imputed cases and controls from a common SNP list. We also created limits around a centroid in the CEU data, the European 1,000 genomes data, where 99% of the CEU sample fell within those limits, and we excluded cases and controls that did not fall within those limits. We also, for the GWAS analysis, used mixed models with genomic relatedness matrices and ancestry PCs. And for this analysis, we wanted to use 2 control cohorts because we were concerned that there would be effects specific to each control cohort, false positive effects. And so we wanted to basically filter out that signal. So we conducted a control GWAS before the case control GWAS, and then any suggestive signal was filtered out of that subsequent case control GWAS. And I'm showing you the results here of the 3,400 suicides and about 15,000 controls. These are the results from the GWAS indicating 6 variants from 2 loci associated with suicide death at a genome-wide significant level. We had another 52 variants that were nominally significant, and those mapped to 19 genes. As you can see up in the Manhattan plot here, the genome-wide significance level is in green, and our FDR correction in purple is at 0.1. And our lambda for this analysis was 1.015. And here are the top SNPs meeting genome-wide significance on chromosomes 13 and 15. Most of these are intergenic. And our LD score regression SNP-based heritability for this analysis was 0.246 with a standard error of 0.036, so highly significant. These are our gene-based results from the GWAS. 10 met FDR correction at 0.1. Through FUMA, we used MAGMA to detect 19 genes associated with suicide out of about 18,000 genes total. Our lambda for this test was 0.994. And associations of chromosome 13 SNPs were found with 3 genes, including DAC1. 11 of the genes, of the 19 genes, have prior evidence of association with suicidal behaviors. And there was a significant association with schizophrenia results from the GWAS catalog. 10 of the genes also evidence significant differential gene expression in postmortem brain in either schizophrenia, autism, or bipolar disorder, and this is corrected at 0.05. So we wanted to leverage the fact that we had 2 waves of suicide death samples, and we had 2 control cohorts to cross-validate prediction of polygenic risk for suicide death on case control status. So what we did here, and to orient you to these plots, on the y-axis you'll see a centered around 0 polygenic risk score for suicide, and on the x-axis you'll see the p-value threshold cutoff for the polygenic risk score, starting at 10% of the variance all the way up to all of the variance. And in the first plot here on the left, you'll see that we used wave 1 and generation Scotland as the training sample to score wave 2 and UK 10K. And then the procedure was reversed on the right-hand plot. And in both cases, suicide PRS significantly differentiated cases and controls. For this next analysis, I wanted to characterize the polygenic risk in the suicide deaths, polygenic risk for different disorders potentially relevant to suicide death. So here, these plots are a little different. These are plotting suicide in red, Generation Scotland and UK 10K controls in green and blue. And these PRS are based on summary data from large extant GWAS of multiple phenotypes. And what we find is significant increases in, in the phenotypes we would expect to be related to suicide, including depression, autism spectrum disorder, disinhibition, schizophrenia, and alcohol use. We also wanted to sort of dig into some of the clinical data and the mode of suicide data because, and there are 4 sort of types of suicide that we derived, because some of the epidemiological research suggests pretty complex etiologies and potentially risk subtypes. So I looked at ICD diagnoses from the electronic health records in the suicide deaths and mode of death as well with respect to polygenic risk for these conditions. And one notable finding I think that's worth mentioning is suicide by violent trauma, which is more rare. It's the more rare form of suicide, was associated with both a clinical diagnosis of schizophrenia and with polygenic risk for schizophrenia. Now, I'm putting up here some of the more prevalent diagnoses associated with in the suicide sample here to show that while schizophrenia rates are very high in this sample, the diagnoses associated with suicide death are typically quite different. So we see a lot of pain-related depression, anxiety diagnoses, as well as cardiovascular and metabolic disorders. So just to wrap up, we have some future goals here with a larger cohort to complete a replication with PGC controls. We are working on a multi-trait-based conditional and joint analysis using GWAS summary data. I'm interested in a trans-ancestry analysis when we finally have enough samples with significant admixture, and we have located, with Dr. Stahl's help, genomic psychiatry cohort controls. We're doing some genetic subtyping with our GWAS, and we are working on leveraging suicide death PRS to start to enhance prediction models of suicide mortality as opposed to suicide attempt with colleagues, Eli Stahl and Niamh Mullins and Ruderfer. With that, I will wrap up and thank all of the contributors to the study and consultants, and I'll hand it over to Eli. Thank you.
Thank you, Anna. We have another short period that we can take questions. So please, again, type questions that you may have into the Q&A box or the chat box, but also you can raise your hand and I can unmute you. We do have one question that was typed into the chat box from Paul Nestat. And if you want to read it and answer it, or I can if you want.
Oh, I don't have the chat. Can you? Okay,
question. Was there a reason that controls weren't drawn from the Utah Medical Examiner's other death investigations, non-suicide? Presumably, if they've been saving blood from suicide deaths, they also did for others. In Maryland, we plan to use MVA passenger deaths as controls when we look at suicide bloods from the medical examiner, for instance.
Right, that's actually, I can answer that. That's a perfect control group, and we have been trying to pursue permissions for that. Our medical examiners have been down by about 4 medical examiners. They've recently hired some more folks, so they're just way too busy, way overworked, and every time I ask them for more, they say, we'll think about that. So we have been definitely working on it. We have a couple of other potential opportunities for some biorepository samples, but we're really, really interested in getting a Utah-specific control group. We recognize this is a big issue. We have a small Utah control group. The unrelated people from the Sept CEU are Utah people. We have another control group that's individuals who are elderly, healthy, study. A lot of the studies in Utah are families because that's kind of our big strength here. So that's a bit of a challenge, but we've been sort of collecting unrelated people from various family studies also as controls. So yeah, we're working on it and definitely working on our medical examiner as well. Lots of, donuts down to the medical examiner, see if they can help us out with some of those samples.
Okay, are there any other questions? I don't see any other questions at this time. So let's move on and hear from Doug Ruderfer.
Excellent. Well, thank you, Eli, for organizing this. And it's happy to be part of this session. I'm glad this deserves a whole session for the worldwide meeting, and happy to have great colleagues that are dedicated to these issues. We came into this for the same reasons that Hilary mentioned, for all the important public health reasons and for the necessity to kind of learn about the underlying biology of this devastating kind of outcome. We came at it from a slightly different direction, and that's partly out of a requirement, and everyone's familiar with this idea that population samples do have limited power for phenotypes that are of low prevalence. And for our own work at Vanderbilt, we have about 3 million unique patients that have been seen at least once at the hospital, but many of those patients are actually come for particular procedures or give birth or because it's a trauma center or go to the ER and don't have a lot of actual data. If we had 3 million unique patients and we had DNA in all of them, we could have a pretty well-powered genome-wide association study even for a 1% disorder, but we clearly don't. If we think about the individuals where we have longitudinal data, multiple events, what we call our frequent visitors or medical home patients, we substantially reduced the number of individuals, about 1 million patients. And of course, collecting DNA in all of those would still be a challenge, though we have a pretty robust collection process where we have about 250,000 individuals that have DNA in our biobank named BioView. But as you see these numbers reducing at this very moment, of course, this is increasing rapidly. About half those individuals have been genotypes. And again, this has changed dramatically over the last year or so, and has also changed over kind of even within this talk that I'm going to give. So we don't really have enough cases in the biobank to constitute a simply powered genetic study. But what we were trying to think about is how we can leverage this information, this extensive clinical information, the clinical data that we have, and the large overall sample size that exists to improve our ability to study the genetics of these low prevalent disorders. And that's kind of the question that led us to think about suicide as a particular phenotype of interest, and one that has been hard to ascertain, but also many other potential phenotypes. And just in the interest of time, to cut to the chase, what we were trying to do here and what our approach was, we think about our sample of 1 million patients. We can identify cases as gold standard, chart validated, best quality possible set of individuals. And then we can utilize the clinical information that we have from our electronic health records. That could be structured data, which is a little bit easier to utilize, or it could be notes or labs of diagnoses, medications, procedural codes to build a predictive model. And the idea is that we can build a predictive model and we can demonstrate that model is both predictive and accurate, meaning that if we say a person's a case, they are a case, and well calibrated, meaning that if we say an individual has 20% probability of being a case, then 20% of those individuals are actually cases. Then what we've done is we've actually converted our binary outcome into a quantitative measure, which theoretically should be well more powered than using case control status. So that's the argument, the theory that we're trying to work behind. There's some really nice work, theoretical work by Yang, Ray, and Bisher, actually doing these calculations and comparing a effective power between a quantitative trait and a binary trait, showing that particularly for low prevalent phenotypes, you have many fold more power to study the quantitative trait than the binary trait itself. And I suggest looking at the paper, it's actually pretty fantastic. This leveraging kind of work that has previously been going on by my great colleague, Colin Walsh, we wanted to apply this to suicide again for all of the public health reasons and for the interest. And at the time when we started this a few years ago, there was limited knowledge in terms of the underlying genetics for this particular trait. In general, it's great to see that that has advanced rapidly over the last couple of years. In reviewing charts of all individuals that had self-injury codes, about 8,000 individuals, they looked through every single one to identify the ones they could confirm by hand, so validate through the charts, that had a suicide attempt. And it was about 3,000 individuals among our data set that had that chart validated suicide attempt. We could build a model now to predict suicide attempt, leveraging the rest of the data that we have, including demographic information, medications, diagnoses, and hospital utilization. And it turns out that using random forests were performed the best among a number of different approaches and provided predictive accuracy closer, the AUC is close to 0.9 or so, and good calibrated model. So fitting the expectation that we think in theory should provide us a quantitative trait that would be well powered compared to our binary outcome. And we applied this to the sample that we had at the time, which is about 25,000 European individuals that were genotyped. And to keep in mind, among those individuals that were genotyped, 73 were cases. So what we're trying to do here is leverage this clinical data to build a quantitative trait to demonstrate that we might have power to do a GWAS on 73 cases using the quantitative traits. So we needed an out group to compare this to. We needed a classically defined patient outcome to do this in. So we leveraged work with our colleague Niamh at Stanford to use the UK Biobank. Among the 500,000 individuals, about 150,000 of them filled out an online mental health assessment and received the question, or the ones who responded yes to a self-injury question received, have you ever harmed yourself with the intention of ending your life, which is a good suicide attempt question because it includes intent. 3,500 of those individuals responded yes. After quality control of the genetic data and reducing to a homogeneous white British ancestry, there are about 2,400 cases of suicide attempt. And we compared to all the controls. If you compare to only the controls that filled out the online mental health assessment, the results do not change. So this provides us now with a classically ascertained self-reported patient suicide attempt phenotype and data set to compare that as well-powered. Although again, neither of these studies should be well-powered enough to a very large scale GWAS for a polygenic trait. So the first thing we can do is actually do a genome-wide association study in the UK Biobank, and not surprisingly, we did not find any genome-wide significant loci. We can then take this result, build polygenic risk scores from UK Biobank, and apply it to our case control status in our BioView sample. And we do see nominally significant enrichment, even though it's 73 cases, that have these individuals carry higher genetic risk for suicide based on a UK Biobank GWAS than the controls. And if we then compare now to our predicted probability of a suicide risk, we see that a much more significant correlation between underlying genetic risk of suicide and our predicted probability of suicide attempt. And this holds even if you remove those 73 cases. So that the power is not generated from those cases, it's actually generated from the clinical data that informs that probability into the controls, as we'd expect. We can do a genome-wide association study in just our BioView sample, and we do see 2 genome-wide significant hits that we've tabled at the moment because neither of them have been replicated in UK Biobank, but are worth investigating further as we increase sample size, which Niamh will tell you more about direct efforts to do that. And more interestingly, we can look at our QQ plots, so we can look at our heritability. We do find both significant and comparable heritability estimates between our UK Biobank sample and our BioView sample, around 4 or 5%. And I think more interestingly, we can do a direct genetic correlation calculation between these 2 samples, identify a significant genetic correlation between the 2 different samples in 2 different ways of phenotyping, close to 1 with high confidence errors. And we also can show that we have significant genetic correlations with traits that we would expect based on our understanding of clinical risk for suicide attempt, including depressive symptoms, major depression, neuroticism, schizophrenia, insomnia, and somewhat more interesting, a negative relationship with age of first birth, which has shown up in other disorders, including schizophrenia. So to kind of summarize, we think that this type of model, again, if you can be accurate, if you can have good calibration, can capture genetic contribution, effectively did a GWAS with a small number of 73 cases, identified significant heritability, and showed that it was genetically correlated, significantly genetically correlated with a standard patient-reported outcome. We think there are lots of opportunities to utilize this approach for phenotypes that are either hard to ascertain or of low prevalence. And we think about both in our work, but also in UK Biobank now that they have primary care information. And the nice thing about this type of approach, again, if it works, is that your power increases with sample size, not purely with case numbers. And we're trying to do this now with the additional 60,000 European samples we have in our own BioView sample to further demonstrate that power increase. We do have work that is currently funded with both Colin and our colleague Roy Perlis at MGH to do this the same approach and apply it to treatment resistance in depression and schizophrenia. And we've, in suicide realm, have been going further with our colleague Adi Bayan, thinking about how do we both identify more individuals with notes. One of the things we're missing currently is all historical suicide attempts that we can capture thousands of more individuals that have historical suicide attempts. We can capture suicidal thoughts or ideation from using clinical narrative notes, and we can demonstrate, we can both improve our prediction model, but also demonstrate the accuracy of that using NLP with the predictions that we currently have. So kind of reiterating improved models, improved power for genetic studies as we move forward. So with that, I'll just thank the wonderful colleagues that in all of the suicide genetics here, my colleagues directly, Colin, Adi, and Manny, everyone in my lab, and of course, all of the funding and everybody else for listening.
Thank you, Doug. Again, let's have a brief question period now. You can raise your hand as an attendee and I can unmute you to join the conversation, or you can type your questions into the Q&A or chat dialog boxes. So there is a question in the Q&A. Doug, can you see it? Or I can read it to you if you want.
I can see it. So, right. So I think that we had started to, so let me read the question in case everyone is not looking at it. The data from Adrian Campos is, what data is missing from the UK Biobank to calculate build a comparable suicide liability model for GWAS? And until recently, much of the data was missing. So it was mostly self-report, patient report information, which we toyed around using, but the extensive longitudinal information becomes really valuable for lots of the model that we're currently using, which is based more on diagnostic codes, medications, and procedural codes, which now, given that I think 43% of individuals have their primary care information, I think it's way more possible to leverage that information to build similar models in the UK Biobank. And that's kind of our hope to move that forward soon.
Thank you. Are there any other questions at this time? Okay, we can move now to our final presentation, Niamh Mullins.
Okay, can you see my slides? And can you hear me? So this is a within case design, and it's aimed at detecting associations specific for suicide attempt and not the psychiatric disorder, or variants occurring at a higher frequency in suicide attempters compared with other psychiatric cases. I ran a GWAS of suicide attempt in each disorder, followed by meta-analysis between them, and in total there were over 6,500 cases of suicide attempt. There were a couple of genome-wide significant associations, one in the GWAS of suicide attempt in MDD. It had a MAF of 2%, another in the GWAS of suicide attempt in bipolar disorder, which had a frequency of 20%. In the GWAS of suicide attempt in schizophrenia, there were no genome-wide significant associations. In a meta-analysis of suicide attempt in mood disorders, there were 2 GWAS hits, one of them on chromosome 2. Again, it had a low MAF and only 1 SNP passing the threshold. But this one on chromosome 4 is the same locus that came up in the GWAS of suicide attempt in bipolar disorder. The association strengthened in the meta-analysis, 10 SNPs passed the genome-wide significance threshold, and there's a nice peak here of SNPs in LD. The region is located in a long non-coding RNA, and in support for our study design, it has not been associated with either depression or bipolar disorder, suggesting its specificity for suicide attempt. So this looked like a very promising locus, and we wanted to try and replicate this association. So we went to our collaborators who work on the UK Biobank sample and the iPsych sample in Denmark. They pulled out cases with mood disorders, split them into suicide attempters and non-attempters to replicate our study design. The numbers were very decent. We had good power for replication, but there was no association between this top SNP and suicide attempt in either of the replication data sets. One of the most interesting findings from this paper was that polygenic scores for depression are associated with suicide attempt in each of these psychiatric disorders. So what I'm showing here is a polygenic score for depression predicting suicide attempt versus non-attempt from left to right in bipolar disorder, MDD, and schizophrenia. So even within depression, for example, depressed cases who made a suicide attempt have higher genetic risk for depression compared with depressed cases who don't make a suicide attempt. And even though this is all PGC data, there's no overlap here between the discovery and target samples. It was also interesting that this finding seems to be specific to the depression polygenic score. So here in the pink bars, I'm testing whether a polygenic score for bipolar disorder differs between suicide attempters and non-attempters with bipolar disorder. There's no difference between the two groups. What I'm showing in green is that there is a difference in polygenic scores for schizophrenia between suicide attempters and non-attempters with schizophrenia, but it's actually the non-attempters who have higher genetic risk for schizophrenia. And as I said, the suicide attempters with schizophrenia have higher genetic risk for depression. So essentially the take-home message from the polygenic scoring analyses is that across psychiatric disorders, suicide attempters have a higher genetic liability for depression, not just a higher genetic liability for the psychiatric disorder that they are affected by. And even though we haven't reached the sample sizes where we're replicating genome-wide significant associations for suicide attempt across cohorts, we are starting to get insights into the genetic etiology of the phenotype using polygenic methods. This was a collaborative effort from many people in the PGC and 3 of the working groups, so I'd like to acknowledge all of the collaborators, and the GWAS summary statistics are all on the PGC website. So for the final part of the session today, we wanted to introduce a new initiative that all of the presenters are part of and many others as well, and that's the International Suicide Genetics Consortium. This is a working group that was set up last year, and our goal is to really scale up genetic studies of suicide to the sample size that we all know is needed. Our initial aim was to conduct a meta-analysis of all the GWAS summary statistics that we could get on suicide attempt or death. We have 15 participating groups, and so far we've gathered data on over 27,000 cases of suicide attempt or suicide, including a range of psychiatric diagnoses, as well as several genetic ancestries. The group was set up by myself and Doug, along with help and direction from Catherine Lewis and Eli Stahl. These are all of our members. All of the studies presented today are represented. We also have the Generation Scotland study, Army Stars. We have 2 African American cohorts. Johnson are working with us, Columbia University, the German Borderline Genetics Consortium, Karolinska. Most of the samples are European. We do have 2 East Asian cohorts as well. Recently, we received data from the new Australian Genetics of Depression study. And of course, the PGC are involved, including the Eating Disorders Working Group. So it's a worldwide effort. You'll notice that we have many different types of cohort here, and we really wanted to try and leverage all of the data that we could for this project. And so we designed the study under 3 analysis models. Depending on the type of cohort, it can be analysed under 1 or more of these models, and the difference between them is really the type of controls that are being used. Model 1 is a GWAS of cases of suicide attempt with a specific psychiatric disorder versus non-attempters with the same disorder. That's a within-case design, the same style as the PGC GWAS. Model 2 is a GWAS of suicide attempt or death versus general population controls, and Model 3 is suicide attempt or death versus screened non-psychiatric controls. And in both Model 2 and 3, the controls are also screened for suicide attempt if that information is available. These are the numbers we have in each of the models. So we've far exceeded the sample size of any of the previous individual GWAS on suicide, which is a great start. We did anticipate from the outset that there would be a difference in statistical power between these models, with greater power from Model 2 and 3, not only because of their sample size, but also the phenotypic difference between cases and controls in these 2 models. One limitation of Model 3, which is the case versus screened control model, is in distinguishing whether any associations are really for suicide or whether they might reflect genetic liability to psychiatric disorders. But we will look for convergence of evidence across the models, and model 1 is designed to distinguish between those scenarios. So we're using these 3 models to leverage all the data that we can, to accelerate discovery and to complement each other. Most of the cases in the study are of suicide attempt. Some are of death by suicide, but we didn't include individuals with suicidal thoughts only in the case group. In terms of the ascertainment of suicide attempt, most of the studies had used a psychiatric interview. All of the collaborators conducted the GWAS in their own sample. It's a within ancestry GWAS under the appropriate model depending on the type of controls they have. We all used a standardized analysis protocol that we came up with to minimize heterogeneity across the analyses. The GWAS summary statistics were shared within the consortium, and we ran a meta-analysis across all studies regardless of ancestry within each of the 3 models. So those are our data. This is our setup. We're quite a new consortium and we have a big analysis plan ahead of us. These are the preliminary results from the 3 models. So there are 6 genome-wide significant loci, all of them from either model 2 or 3. There is significant SNP heritability in model 2 at 6%, 13% in the model of cases versus screened controls. In model 1, there were no genome-wide significant SNPs and only a hint of SNP heritability, which was non-significant. So as we anticipated, model 1 likely has the lowest power out of the 3. The gene labels here are just the nearest gene to the index SNP. Some of these regions have been associated with psychiatric disorders before, like the MHC and SHANK2. So we have secondary analyses underway now to really delve into these loci and to dissect these results further using polygenic methods as well. So watch this space. We have a presentation next month at the World Congress of Psychiatric Genetics. So we're looking forward to sharing more results then. And we're also really excited that there's an entire session on the genetics of suicide for the first time. So I think that's a sign of all of the work that's going on in this area. These are all members of the ISGC. We've all been working together over the last year or so to get this study up and running, and if anybody else would like to get involved, feel free to send us an email.
Thank you, Niamh. We'd like to take any questions, perhaps initially specifically for Niamh, and then opening it up to any of the panelists. There are 2 questions. Again, please raise your hand as a participant or attendee, and I can unmute you to join the conversation, or you can type a question into either the Q&A dialog box or the chat dialog box. So there are 2 questions. One is in the chat box from Dave Curtis. And Dave, shall I unmute you and allow you to ask your question? Let's see. I believe I'm trying to unmute you. I'm not able to unmute you.
How about now?
Yes.
Can you hear me? So
Yes.
it's just generally an idea about you have people with diagnosis of depression, and you say some have a suicidal attempt, some don't. But although they've all got depression, it might be that the attempters have more severe depression than the non-attempters. And so what you may be picking up is not really suicidality, but kind of general depressive severity. And then if you take in a different diagnosis, schizophrenia, you may have some people with schizophrenia who tend to be more or less depressed, and the more depressed ones tend to be more suicidal. But again, it might, I'm not really sure if we're picking up something specific about suicidality or whether it isn't other features of psychopathology, like how generally depressed somebody is, how generally disinhibited they are, how impulsive they are, how borderline they are, how substance abusive they are, and all those other quantitative traits that we would expect to vary with between people within the same diagnosis?
Yeah, so that's definitely what we're seeing across the polygenic scoring, that suicide attempters always have higher genetic risk for depression regardless of their psychiatric disorder. And that is fitting with the clinical picture. So for example, the presence of depressive symptoms in schizophrenia are a risk factor for suicide. Longer depressive episodes in bipolar disorder are a risk factor. And we did find in the PGC data that the suicide attempters with schizophrenia did have higher factor scores for depression. So yes, it's fitting with the clinical picture. Our analyses, though, were aimed to try and tease apart the genetics of suicide attempt compared with the psychiatric disorder. And so I can pick up associations specific for suicide attempt, but also given what we know about the polygenic nature of these traits and polygenicity between many disorders, we don't necessarily expect all of these genetic variants to be specific for suicide attempt, but we'd expect to see some overlap with other disorders. And the within case model in depression, for example, not only can it pick up associations specific for suicide, but associations that are pleiotropic with depression but occur at a higher frequency in suicide attempters. So what we've seen from this study and many others is that the disorder that suicide attempt seems to be most closely genetically related to is depression.
Any follow-up?
No, that's fine.
Let's see if I can reject you. So the question in the Q&A comes from Adrian Campos. If Adrian, you would like to ask your question yourself, I can allow you now to unmute yourself if you would like.
Yeah.
Please go
sure.
ahead.
Thank you. Can you hear me?
Yeah, please ask your question as well as, yes.
Yeah, I was just wondering whether there was any evidence for differences in inflation or power between different types of ascertainment for a suicide attempt. For example, you mentioned you had some groups with psychiatric interviews versus self-report or medical health records.
Yeah, so this is in the ISGC study. We do have several different methods of ascertainment. Most of them are psychiatric interviews. Some studies which did online recruitment used self-report. Some studies used hospital records like ICD codes. So yeah, there's possibly some heterogeneity in the definition of the phenotype from different methods of ascertainment. We do intend to look into that. So for example, we could look at the genetic correlations between the different cohorts depending on the different methods of ascertainment. And we'll certainly look at this. One challenge with doing that is though the statistical power and the sample size to do genetic correlation analyses from each of the cohorts individually. It does pose a challenge to testing that.
Yeah, all right, thank you.
Thank you. We are at time. I'm not sure what happened at 11 o'clock, but if there are any other questions, perhaps there's just a tiny amount of time remaining. And if not, I would like to thank you all again for attending the PGC Worldwide Lab Meeting, Insights into the Genetics of Suicide. Thank you. Have a good day.